Welcome everyone. We'll just wait a minute or so for people to log on. We've currently got 7 people with us, so we'll make a start very shortly if you don't mind holding on the line there. Okay, excellent. Well, we might make a start. We've got about 10 people that have joined us already, so welcome Andrew, Andy, Angelica, Fiona, Ian, John and Richard. We'll get others on the line shortly. This webinar today is being delivered as part of the Integrated Crop Protection Project, which is a national extension project aimed at extending existing research and development in the vegetable industry. We're very lucky to have Paul Horne and Jessica Page with us from IPM Technologies today, who are our project partners along with Applied Horticultural Research. My name's Carl Larsen from RMCG and I'll be facilitating today's discussion. Before we get into the presentation and I hand over to Paul, I might just run through a bit of housekeeping. We've got a number of handouts on the sidebar there which should show in your control panel. This is our Mega Pest series, which starts with the basics of protecting your crop and then moves through to different types of pests, including chewing and biting and sucking pests, as well as soil-borne diseases. So feel free to download those now or at the end of the presentation. They'll also be emailed to you afterwards as well. We're lucky enough too to offer anyone that joins the webinar today to receive 1 month's free integrated pest management advice from IPM Technologies, which is great. I'll be emailing some details about that after the webinar to you, so thanks very much for joining us. I'll hand over to Paul Horne now, who will be walking us through the presentation for about 30 minutes, and we will have the chance for questions in the remaining 30 minutes as well. So feel free to type questions into your question bar there on the control panel on the right-hand side. We can see those come up at any time and we'll address those at the end, which would be great. Paul, over to you.
Okay. So the idea of today is I just run through some of the basic principles of how you control pests in general and some things that may not be obvious about how pest problems can be made either worse or better. So as Carl said, this should go for around about half an hour and then Jessica and I can answer any questions that people want to put to us. So we believe that there's really only 3 control options for any pest in any crop. Biological control is the first thing that we list, and that is the beneficial insects and mites, the predators and parasites that eat the pest insects and mites. Cultural controls is a much more diverse and sometimes quite difficult to understand and to get around, but it's really the management options that farmers have available that have an impact on either the pest, so making life worse for the pest, or favouring beneficial species. So they can be extremely powerful and will be the topic of one of the later webinars in a month or so, but they're often very overlooked. The third control option is chemicals, and chemicals have been the basis for pest management and in vegetables and many other crops for a long time. So they're the 3 control options, and I'll just go through some of those and talk about how we can integrate those and use them as an IPM strategy. So in our little company, we work in a vast range of crops, from intensive horticulture through to broadacre agriculture. And it's amazing how similar the problems are and how similar the options are for controlling pests. So where pesticide use might be the starting point, it really is just one of the tools, and we find that we have to encourage people to look at the biological and cultural options and not just stick to chemical controls. So if we look at the three strategies we've got up here, if we say the starting point for many people is a chemical-based or spray-based strategy, that's okay. And then IRM strategies are a further development of that. So insecticide resistance management strategies are still chemical-based, spray-based strategies, but where in an attempt to avoid resistance, people are encouraged to rotate through the different chemical groups and so not to overuse one product. However, that can mean rotating through insecticides that are not compatible with biological control. So an IPM strategy, as opposed to an IRM strategy, has a smaller set of chemicals to choose from. So integrated pest management means integrating what few chemicals there are and not relying on them primarily, but using them as support tools. So this is a photo of a cabbage crop from where Jessica and I have worked for many years in Werribee South. So this crop was sprayed with insecticides. They had a chemical-based strategy. And the holes in that cabbage, and that cabbage was pretty typical of the bulk of his crop, was caused by diamondback moth, of course. So diamondback moth, the caterpillar there, is resistant to a great number of insecticides. So there's only a few still that will work. So again, insecticide resistance will be the subject of a later webinar, so I won't go into it too much. But the point is, this farmer with his cabbages was growing a crop that was unsaleable. So it was a spray-based strategy. He was using 4 insecticides a week, sprayed twice, 2 tank mixed at a time. And of course, if he's using or relying totally on a chemical strategy and eliminating biological control agents in the process, then failure was certain, because he had no biological control, the chemical control didn't work, and so the pest was given free rein and all the food that it could eat. Diamondback moth was the first pest in the world to get resistant to synthetic chemicals, and the first one was DDT. Another example, just to emphasise what happens when insects get resistant to all the insecticides that are available. We encountered a problem in the strawberry industry in Victoria a few years ago. So in an attempt to produce bright red strawberries like the one on the left, Western flower thrip became a problem that was uncontrollable. Exactly the same as diamondback moth. The insecticides didn't work. The fruit in this case was still damaged, and so that the 2 strawberries on the right are scarred by thrips feeding damage, and they basically had a crisis, and the industry had to change and get away from that total reliance on insecticides. So the Western flower thrip was able to tolerate the insecticides used in strawberries, but those insecticides killed all the natural enemies of the thrips, and so the result was damaged fruit and, on occasions, residues, unacceptable residues in the fruit as well. We also have to deal with insects, not just because of the direct damage that they cause, but because they can vector viruses. So the photo here shows tomato spotted wilt virus. This can be spread by a few species of thrips, including Western flower thrips. So in the previous example with strawberries, it was direct feeding damage only, but in other crops, the same pest that's insecticide resistant can also vector disease. More recently, a problem for growers has been trying to deal with insects as contaminants. So Rutherglen bug in this photo, some years are worse than others, but when they fly into crops such as lettuce or salad mix, they can cause problems and have product rejected by buyers simply because of their presence, not necessarily because of any damage to the product. But all of these things are pests that have to be dealt with, and we have to deal with them all in some compatible way. So we encourage people to use IPM, of course, and the reasons for doing that, some of them are listed here. So I've already talked about the first one. When the pesticides stop working because of resistance, growers are more or less forced to do something different. But pesticides are also withdrawn. They can be withdrawn by government regulation, but they can also be withdrawn, and it has happened recently with some insecticides where the company that previously sold them doesn't want to produce them anymore. If product is to go overseas, then the export requirements have to meet the country that they're going to, not just Australian requirements. And so it could be quite possible to grow products using chemicals that are legal here, but they're not acceptable in some countries overseas. So the grower has to take that into account as well. I'm going to talk a little bit more about pest flare and give some examples where choosing the wrong insecticide actually can make pest problems worse. Residues in produce, that's obviously an issue. The product has to be within the regulations and achieve MRLs. And of course, worker safety. Some of the older insecticides, the organophosphates in particular, were highly toxic, and if people didn't have to use them, then usually they prefer not to. So I've got a graph here where I'm going to try and explain how the wrong insecticide was used and it actually made problems worse. So what it shows is counts of aphids on potato leaves. And we worked with a group of farmers, and only one of that group insisted on using an insecticide because that grower was growing a seed crop and didn't want to tolerate aphids because of the risk of virus in the final product. So all the other growers decided that they would agree with our recommendations and didn't use any insecticides whatsoever on the crops that season. So the three arrows point to where the seed potato grower sprayed 3 insecticides. And the red bars show the number of aphids in his crop. Now around week 8, which is midway through the season effectively, there's a little yellow bar which shows the number of aphids in the unsprayed crops. So what he did was actually use a broad spectrum insecticide that killed all the beneficials in a potato crop, and the canopy in a potato crop is obviously quite dense from the midway on. But it didn't kill the aphids. The aphids were on the underside of the lower leaves and were probably able to tolerate the spray that was used. It was a broad spectrum, not a selective one, and with the short generation time that aphids have in the warmer months, basically there were 3 generations of aphids without biological control or cultural control, and so that's an example of pest flare. And so this is back to the cabbage crop in Werribee South. The ones that weren't full of holes were full of cabbage aphids, and this was before Movento was available, and there were no insecticides that could control the aphids inside a cabbage at that time. So all the grey colours there, they're all cabbage aphids. I'll just give one more example, that's one that I found quite interesting, where a farmer, again a potato farmer, wanted a crop sprayed by plane, and he wanted a fungicide put on, and the fungicide was called ACE. But when he sent the email, he actually requested AXE, and unfortunately for him, AXE and ACE are not only both words, they're both pesticides, but one's a fungicide and one's a synthetic pyrethroid, so a broad spectrum insecticide. And by the time the mistake was realised, half a paddock was sprayed with AXE, the other half was not sprayed with the synthetic pyrethroid. So we looked at the aphid counts in that crop on either side of the paddock. So this graph shows the percentage of leaves with aphids before and after that insecticide application went on. And if you look at the blue line, that's the half of the paddock that got sprayed with AXE, and it took about 4 weeks to go from effectively 0 to 100% of leaves with aphids, whereas the other side of the crop was actually hardly any change. The difference between those two lines is a measure of the biological control that occurs naturally until it's disrupted. And so the point here is that it is often assumed that if I put an insecticide on, it can't do any harm. So we put it in just in case. So I think this shows that if you choose the wrong insecticide, it actually does make a problem far worse than it would otherwise be. So in all crops, it's very simple. If you lose the beneficial species, and that can be achieved by a pesticide targeting one pest, and it may control it, but it will induce the flare of another pest. So for example, you can put an insecticide on for looper caterpillars, and it might control the looper caterpillars but induce aphid flare. So if we kill those predators and we induce new pests, quite often those pests are tougher because they've already survived the sprays that have been applied. So if I talk a little bit about beneficial species, so the biological control agents in this case, not just things like pollinators or worms, so the biocontrol agents are either predators or parasitoids. So predatory things like ladybirds and lacewings, and parasites, mainly wasps, but also fly parasites can be quite common. And there's one more difference in how we can separate them. They can be either resident, so living in the paddock, so they're permanent inhabitants of that piece of land, or they're transient, which means they move in from outside the cropping area each season. So things like lacewings wouldn't be resident permanently, but would move in from either native or other crops nearby. I'll show some pictures of the different species of pests and beneficials, and because if we accept that for IPM you're going to be relying on biological control, then identification of not just pests but biological control agents is going to be important, and misidentification can be really significant. One example we had years ago was where people were not recognising that they had earwigs as a pest and applying slug bait, and in fact they were just completely wasting their time and money. So I can show some photos now of somewhat what we think are the key beneficials in vegetable crops, and the first one is brown lacewing. So the brown lacewing adult is about 8 or 9 millimetres long and lives in a whole range of both natural and cropping systems where it's low growing vegetation. So it doesn't matter if it's wheat or barley, or potatoes or celery, it will live in low-growing vegetation where there's appropriate food. And food that it prefers would be aphids or very small caterpillars, things like that. So the adult flies into the crop and lays eggs on the underside of the leaves. So those 3 eggs shown on the left. The larvae hatch out, and they're very voracious, and so the photo on the right shows a brown lacewing larva with an aphid in its jaws. It has 4 hollow jaws, and so essentially it just sucks the insect dry. We also find hoverflies at various times of the year that are very abundant and also have a big effect on aphid colonies. Particularly, we've seen them have a huge impact on cabbage aphid in brassica crops. So this photo shows an adult hoverfly feeding on a flower. They're not predatory in the adult stage, but they're predatory as a juvenile. And so in this photo, there's a whole lot of aphids, which are food for brown lacewings and hoverflies. So the large maggot in the lower middle of the photo is a hoverfly larva, and the brown lacewing larva, for comparison, just above it. So both of those are very important predators of brown lacewings, of aphids, and they can be extremely abundant during the spring and early summer. Later in the season, we start to find damsel bugs, and so this is a true bug. It's a sucking insect, but it doesn't feed on plants. It feeds on other insects, particularly small caterpillars. So this is, again, one of the most important species that we see. All these occur naturally. They don't need to be bought. They're not reared commercially. So they all arrive and they do an immense amount of biological control in vegetable crops. Now, this photo shows both a predator and a parasite. So the large maggot on the right is a hoverfly maggot. The winged insect is an adult female wasp, and it's in the process of stinging an aphid. And when it stings the aphid, it puts its own egg inside the body of the aphid. That hatches inside the aphid, turns into a maggot. The maggot eats the aphid from the inside, and then uses the skin of the aphid to form its own pupal case. So this is what they look like at the end of the process. So these were aphids, but they're now just cases containing wasp pupae, and an adult wasp with wings will come out of each one of those. The important point is that whereas you can see juvenile predators and you can see them working, the juvenile parasites are hidden, or the parasites are hidden for the bulk of their life as they are inside the body of their host. So probably only a few days before this photo was taken, those would have looked like quite healthy aphids. And so it's more difficult to see the parasites at times than to see the impact of predators. So this is a particularly good parasite for controlling diamondback moth, and that's one called diadegma. That's the female with the ovipositor. There's a little prong out the tip of the abdomen. Some others that are native species, the one on the left was a looper caterpillar, but it has been turned into basically a bag of about 1,000 wasp pupae in that photo. Similarly, the photo on the right is a cutworm, and each one of the little cells that you can see contains one wasp. So there'd be more than 500 wasps in that body of the caterpillar. So these occur naturally, and they're underestimated in most cases because we see the caterpillars, but we don't wait often we can't tolerate them until the parasites actually take over the body. Just talk very briefly about cultural controls. This is again the subject of a webinar later on, so I won't go into it too much. But some of the management options that we have that are very powerful include variety selection. So if you've got a resistant variety, so perhaps lettuce that's resistant to lettuce aphid, then that's all that is required. There's no need for biological or chemical control for lettuce aphid in that case. Irrigation, overhead irrigation can be used as an important control in some crops. Weed control to make sure that there's not a carryover of pests or a build-up of pests is often, you do it for other reasons, but weed control is one of the most important cultural controls for a range of things. People often ask about border plantings. What can we plant to encourage beneficials? And we'll probably leave that for another time, but we can either have trap crops or things to encourage beneficial species by providing a nectar source at the edge. Getting to the chemical controls, we see that chemicals should be used as the support tools, not the mainstay of control of pests. And so the selection of what product should be used needs to be based on the impact on beneficial species, not just on the pest species. So we're finding that compared to 10 years ago, there's more selective products than ever that are available, but if we overuse them, if we use them as standalone products and not integrating them with biological or cultural control, they'll be overused and will get resistance. So it makes sense to us to get more value out of those selective products by using them in an IPM strategy. The harder products, the older products, the organophosphates, synthetic pyrethroids, can still have a place if they're used in, say, baits or as border sprays. So they're not used on the actual crop, or they're only going to target the things that eat those particular baits. So the three measures that I started off with, the biological, cultural and chemical control, using them together is what is IPM. So instead of relying on one, especially chemicals, we see that chemicals become the third support tool, and the decision about what product to use and when to use it should be based on monitoring of the crop, and monitoring not just for pests but also for beneficial species. So I'll just finish off with an example of how we see an IPM strategy that could be developed. So let's say this is a brassica crop. We look at what range of pests there are in that particular crop. So I know there's more, but we've just said, let's think we've got diamondback moth, cabbage white butterfly, aphids and thrips. How do we deal with that range of pests? So there's a range of things that eat them. So wasps will attack 3 of those at least. Damsel bugs are predators of the small caterpillars, cabbage whites and diamondback moth. Brown lacewings, hoverflies are very important in aphid control. And for thrips, we have predatory thrips and predatory bugs. As far as cultural options, what growers do ordinarily is one important control factor, cultural control. So sequential plantings. So if we build up a population of beneficials, they will move through from planting to planting. Controlling flowering brassica weeds is an important control because it reduces the number of eggs. Sorry, if diamondback moth or cabbage white get a nectar feed, they lay more eggs than if they don't. So controlling flowering brassicas is an important control, as it is for aphid control. Those three top species, that's diamondback moth, cabbage white, cabbage aphids, only feed on brassicas, and so controlling brassica weeds is important. Thrips, sometimes thrips come from adjacent crops, so just simply where the new brassica crop is planted can make it extremely susceptible, say if it's on the south side of where people are harvesting either other brassicas or things like onions at times. The number of thrips moving from one crop to another can be significant. Then some chemicals. Which chemicals can we use that are compatible with the range of beneficials that will still have an impact on those pests? So these are just some examples. There's more that we could use, but for the two caterpillars, BTs, so dipels, entari, things like that. Some of the group 28s for diamondback moth in particular still work. There's selective aphicides that can be sprayed for aphids, and if a spray was required, I think chemicals like Success Neo will have an impact on thrips. So they're just an example of how the three control options are going to work together and not interfere with each other. So I think that's the end of my presentation. I'll hand it back to Carl.
Excellent. Thanks, Paul. That was a really good overview of what options we do have available to us to control pests. I'd now like to open up to everyone that's attending to raise your hand or type some questions into your control panel. We do have a couple to start, though, from John Duff. Now, it was back in the chemical control section that John raised a question about. He would have thought that primer would have been worked on the cabbage aphid and is selective to aphids. Was this used in that particular example?
I think that example was in a potato crop. Oh, sorry, which example is that? Or was that the one of in Werribee, perhaps?
That's fine. What we might do is take that on notice.
I think, John, if it was the cabbage, Pyramore would have worked in the earlier stages. It would work on cabbage aphid. It's just that when they got right into the crop, Pyramore wouldn't get in that far. So when they were entrenched inside the cabbage, they couldn't get it.
And he may not have been using it at that stage.
Yeah, and also that photo was taken before Jesse and I were working with him. So we know he was mixing a lot of different chemicals. I'm not sure whether he would have used Pyramore.
Thanks for that, John. And John's also got another question there about how do you feel about some weeds for parasitoids to use as refuges and food for the adults? What do you think?
So we did some work on that years ago. Jesse and I found that some weeds were definitely beneficial, and so things like aniseed weed didn't seem to harbour many pests, but did provide a nectar source for a range of beneficials.
That was next to a brassica crop.
Yes, that was next to a brassica crop.
Yeah.
And maybe one from the panel, Paul. You mentioned some of the key benefits of transitioning across to an IPM approach. I mean, what do you see, and Jesse as well, in some of the main barriers to growers adopting IPM in the first instance, or advisors as well, beginning to provide IPM advice?
So I guess the thing that we've seen over the years is that while the chemicals still work and they're registered and they're legal, and that's what people are familiar with, then there's little incentive to change. So unless there's a crisis or people have a particular desire to change, then people mostly will still do the same thing.
We've also got another question from John, and John, I might actually try and just quickly unmute you so you can ask those couple of questions. Just hold on one moment. Are you there, John?
Can you hear me?
Yes, I think we've got John there. There was a couple of follow-up questions from the first couple there. Did you want to just make sure we've rounded out and answered those for you? You're on the line and we can hear you.
Yeah. With that cabbage aphid one, I'm just interested to find out, was the grower actually monitoring his crop before you guys took over, or was he just spraying on a calendar spray, which caused this problem?
I think he was just spraying on a calendar spray or weekly spray.
Yeah.
Twice a week. Yeah.
Twice a week, my God.
Yeah.
And the other question I had, I thought success was only good against western flower thrips and not some of the other thrips species.
I think it's actually the other way around, John. I don't think it works against western flower thrip at all in most crops anymore.
Yeah, it used to work,
Yeah.
but they're now resistant to it, in the crops that we look at.
But it works quite well against plague thrips and onion thrips.
Yeah.
Okay.
So, John, does that answer your couple of questions there? I just thought, given that you typed a few in, we might go to you on the audio line.
That's cool. That's good. Thank you.
Oh, great. Thanks, John. Look, I'll just mute John there. We've also got a couple of other questions that have come through. One from Andy Gulliver over in Western Australia. Asking, many growers still use the weekly sprays for diamondback moth control and cauliflower. Are you saying that we can develop an alternative?
I guess we recommend sprays for diamondback moth in the peak time of year, and that could be once a week. So it's just what is used. Our experience with using releases of diadegma, which was the wasp we showed a picture of, with products like BTs and a group 28, we reduced that down to less than one spray a week.
Yeah, I guess it depends a bit on where they're flying in from. So there will be a period where you do spray weekly, but towards the end of the season it should drop off.
Yeah. When that initial movement from broadacre canola, probably when the canola dries out and all the diamondback moth flies in, then it's intense. But if we can we can drop it back after that.
And Andy's also just raised another question. Andy, I might go to you and unmute your audio, just about a question around typical monitoring programs. Andy, are you there and you're on the line?
Yes, can you hear me?
Yeah, we can, thanks. You want to pose your next question around the monitoring program?
Yes, it's a long time since I've done infield monitoring, but I can remember going through paddocks with my ×10 magnifying glass and my notebook. I'm just interested to know what would a monitoring program look like now? Would we go in weekly or daily, and what exactly would we be looking for?
Right.
And I'm happy to focus on diamondback moth.
Oh, I think, well, weekly would be, we think weekly is enough at the peak time of year.
Yeah, we think weekly for several reasons. If we want a grower to do something, they're unlikely to if we came twice a week, they wouldn't be able to get on and do two things in the week. And there's not enough time to see the impact of what actions have been taken. So yeah, once a week, and looking for pests and beneficials with our 10 times hand lens.
So did you have anything else to add to that at all, Jessie?
No, that's pretty much.
No, that's good. Andy, does that cover off on your question?
Another detail, if let's say we're planting, say, about an acre, 4,000 square metres of cauliflowers a week, how big a sample, so that's, say it's 10,000 plants, how big a sample would we need to take to be representative of that planting?
Yeah, so we think an amazingly small amount, really. Unless you have particular reason to think that the pest is coming in on an edge or there's some hotspots, we think that all of the planting are equally attractive. If there's an edge effect, looking on the edge is probably a good option anyway, because that's where pest pressure will be higher. And so we would look at about 50 plants, and it doesn't matter whether we're unless the plantings are absolutely enormous. Typically, when we visit a crop, we would look at a young plant, medium-sized plants, and one getting towards past buttoning anyway. And so covering the range of ages and looking not just for what pests are there, but what has been done last week. Did it work? What beneficials are there? What's the trend with parasitism? And so build up a picture. So the weekly monitoring, I think, gives you a pretty good idea and keeps tabs on it.
Great. Well, Andy, thanks very much for those couple of questions. We might go to Mike Buchy now, who's asked, we have a spray-free vegetable garden and have used Thuringiensis on brassicas with success. How practical is this on a paddock scale, and does resistance occur?
Yeah, it's certainly, both Dipel and Zentari are the basis for our advice to growers in Werribee Cranbourne areas where we do regular monitoring. And it certainly is practical on a paddock scale. There's particular things that you need to be aware of to make it work. And perhaps if you want to contact us afterwards, it'd be more appropriate to go through that. And does resistance occur? Yes. Resistance to BTs has occurred in some of the Southeast Asian countries in the past.
So, Mike, I just might quickly unmute you there, and you're on the line. Did that cover off on your question?
Yeah, that did. Thank you very much.
No.
I just wondered, can you hear?
We can, yeah, excellent.
Yeah. I just wondered whether also the Thuringiensis affects any of the beneficial insects.
No, it's actually very soft. It's safe to everything.
Yeah.
Only kills caterpillars.
Yeah, so Dipel Zentari will only kill caterpillars. There are other strains of BTs, but you really have to hunt for those, and some of them aren't available in Australia. There's strains that kill flies, there's strains that kill beetles, but the ones that we have access to only kill caterpillars. And the question about resistance, Zentari is a different subspecies than Dipel. So there's Bacillus thuringiensis kerstaki, which is what most products are. Zentari is Bacillus thuringiensis aizawa, and so it is, and they have different toxins, and so using both of those, not together but in rotation, does actually help to reduce the resistance issues.
Oh, that's fantastic. Just a last point is, living in Tasmania where it rains, well, often rains quite a lot. You have to apply Thuringiensis every time after it rains. Is that correct?
Well, we hope that we give it 24 hours to work. So if it's working alongside beneficial species, unless there's reinvasion, it should have done the job after one application. So you shouldn't need to reapply it if it actually has worked that first time.
Thank you. That's great. Thanks.
Well, Mike, thanks for those questions. Keep those coming in. There's been a great mix of questions from everyone already. But Paul, I might go back to the panel that we've got here around some of the key first steps in either adopting IPM or where to start out. We quite often get those questions in the field. I'd be interested in the top 3 tips for those looking to implementing or providing advice on this.
What do we reckon?
Start at a time of year when it's not, when the least pressure time of year would be a good time to start. Maybe pick one paddock and not try and change the whole farm all at once.
Yep.
So little steps.
Yeah.
So it's not such a huge risk, or doesn't appear to be such a huge risk. And get some advice, get some help, someone to help.
Oh, that's great. Really practical.
Yeah.
Yeah, certainly seeing is believing, and so the only way is to try it on your own farm or in a real situation, a commercial situation, to get the confidence that it will actually work.
And just on seeing some of those success stories we've done as part of the ICP project, some of those video case studies on learning from grower experience, and some of those on the website integratedcropprotection.com.au do take you through the journey of what others' experiences have been. Trialling in a fish tank town in Cranbourne in Victoria, through to other examples in other states around the country. We've got a question that's come in from Andrew Manners. Thanks, Andrew, for that. Do you recommend growers to count actual numbers of pests or predators, or do you think that qualitative measures would be sufficient? For example, low, medium, and high.
Low, medium, high.
I think low, medium, and high is a good practical way of doing it.
Yep, that low,
it's
medium.
Quick and yeah, if you're doing it weekly, you get a good understanding of what low, medium, and high looks like. Yeah,
and so whatever tolerance one farmer will have, that low or medium or high could be very different on the farm next door. So as long as it's meaningful for you, that would be enough.
Andrew, I might just quickly unmute you to make sure we've covered off there.
Yes, I'm here.
Yeah, great. Does that answer your question, or did you want to go into a bit more detail at all?
No, that was fine. It was pretty much what I was thinking, is just relating it back to the graphs, which were all sort of in numbers. I thought that just wanted to sort of elaborate a little bit.
Yeah, those graphs I showed, they were things that we did because we had the opportunity, and so we could get some numbers, but basically, we follow the same sort of procedure. If the tolerance for a pest is very low, then, you find a couple of pests and, you're already into the high level.
Absolutely.
And have you found a particular situation works better for you, Andrew, either the qualitative or quantitative, or are you trying some different approaches out?
Well, I was more thinking just from your opinion and your experience when you're interacting with growers, because as an educator myself, we've been using or suggesting that growers use the low, medium, high approach because it's a lot easier and faster. Just wanted to
we think that's it's a good way, and also if we often describe what's the threshold. If we've got half a dozen aphids and 2 ladybirds, well, we probably don't have a problem. But that half a dozen aphids without any biocontrol agents, and in a couple of weeks we'll have a massive problem. So really, it is important to look at both, not just the.
But it's also really important that the monitoring is practical and easy. It's more important that it's done quickly once a week than maybe, once a fortnight and you get a much better picture.
Yeah, we don't want the monitoring record keeping to put people off.
Absolutely, and that's the struggle that we've been encountering ourselves.
If it's too odious, no one will do it, and the spray option's just easier. So
absolutely,
IPM has to be to give equal or better results, and it has to be done in an easy way.
Well, Andrew, great. Thanks for that question. We've also we'll duck back over to WA. Andy Gulliver's just raised another question. Now that's a good question. What might be the threshold for diamondback moth where do you start? Which is a really good question.
Well, it's tricky. We don't really use thresholds, but I suppose it depends on the size of the crop, where it is, weather,
so
a whole lot of things.
If it's, you can imagine if it's hot weather, and we're talking about the buttoning stage of broccoli or cauliflowers, and we're finding, 1 or 2 caterpillars on every one and no beneficials, well, then that's a high risk. But if we're only finding eggs and there's a lot of beneficials there and we think they're all going to be eaten, then the pressure is much lower. So we, as Jesse said, we don't like rigid thresholds. We really just try and look at risk assessment and what we can do about it.
Now, Andy, you've had a couple of subsequent questions around is it number of infested leaves or total number per hectare. I might just quickly unmute you to go back to you. Have we covered off on that, or would you like to go into any more detail while we've got you back on the line there?
Sorry, I think you got the wrong person.
Andrew Manners. Sorry, wrong Andrew. Andy Gulliver. Sorry, we should have you back on the line.
Yep, back again. Yeah, that was a good conversation. I get it. You really got to start, and then you've always got to learn by doing to set your own thresholds and understand your own crop and situation.
Yeah, exactly.
Yeah. And I mean, the threshold, if you have a rigid threshold, it also depends on what the price is for the produce this week. Is it worth doing anything about it, or is it, the week before Christmas and everyone's the price has gone through the roof? Thresholds will be dynamic.
I just wonder if there's anyone out there, and maybe the panel as well, who you start with a sceptical grower and you say, look, I want to help you actually introduce IPM, and we're going to start counting stuff and telling you what's there. And I'm just imagining the looks and the comments I'm going to get. I wonder if you can share some of your experience of how you overcame that scepticism.
Excellent.
I think you just encourage them to come out with you each week and then make a decision each week. So the decision is, can it wait another week, yes or no? And just take it week by week and encourage them to make the decision with you.
Yeah, so that was our experience with intense vegetable growing in Werribee. In the initial instance, the growers were so concerned that they would come out with us, they would look over our shoulder, but then once they understood how the decisions were made, it became much more relaxed and just leave it to it. So it's really just about confidence, and as we said previously, if the monitoring is going to put them off because of all the record keeping that might be required by some people, it's not necessary. We've got to make it as simple as the alternative, which is spray this at the label rate. So all IPM turns into is, if we're going to spray something, well, what is it and when do we spray it? So all the biocontrol agents are naturally occurring. The decisions about varieties and so on has already been made. So in essence, the decision making is still the same. So what do I spray? Do I spray anything? And if so, what?
Yeah, and look, I get what you're saying. I'm with you. And of course, we've got to break a habit, because currently the easy thing to do is just go and spray every week, because I can plan it, I can schedule it, I don't have to actually think about it.
That's right.
Yeah.
So I guess you just work with people that are interested in working with you. It's not something that.
We don't push people to do it.
Yeah.
And we've got the comment there from Andrew Manners that was baby steps, and I think that's a pretty good summary of that discussion, is that it is about incrementally seeing that change.
Yeah. When growers see it on their own farm, then they believe, and when the apparent complexity of IPM is taken away, if growers think they have to be able to become an entomologist in order to do it, that's not going to work. They really just have to trust that it's actually pretty simple, as long as they don't disrupt it.
Yeah, just try it on one planting or one farm or one block.
Yeah. Well, Andy, thanks very much for that. We might just mute back. And just off the back of that question around incremental steps and seeing those practical benefits, I mean, when things do go wrong, looking at changing over practices on farm, I mean, what are some of the common causes of that, and how you've addressed that in the field with some growers and advisors?
Well, I suppose because you're making a weekly decision and it's will be what spray to use, there's no reason. I mean, you can go back to spraying whatever you want. If it seems too high a risk, then So that would be.
That's it. One of the risks that we see is if things like the tank, if people are trying IPM on one crop but using broad spectrum insecticides on another, if the tank isn't cleaned out or washed out, next time they come and spray a BT, but it's still got some synthetic pyrethroid in it, that can be a problem.
Or maybe not, and also not using a big enough area to trial.
Yeah. Right. Okay.
It's got to be a decent size.
And when you say decent size, Jessie, what's the kind of minimum threshold that we're talking
about? I guess it's got to be at least a spray tank.
Yeah.
A tank's worth.
Yeah. It's got to usually a planting, monitor a planting.
Yeah. Okay, that's good. Well, look, we've really getting to the end of the hour here. I want to make sure that we do stick to time. If there weren't any final questions, I think, look, thank you very much for those questions. I think it's been a great discussion. As we mentioned, there are a series of 4 webinars, this being the first one, and I'll just flick through to our next steps. Our series of 4, we're going to be covering off on resistance management later in the year, in October, with Paul and Jessie, before we launch into 2017. And I know these are out on the horizon, but we'd encourage you to jump onto the website, integratedcropprotection.com.au, just to register those, and it'll send you the reminders when necessary in the new year. We'll be covering off on the effects of pesticides on beneficial insects, which is a real key question we get asked in the field out there with growers and advisors, as well as the 4th webinar. They're really delving into the cultural control options and some of the options that are there for people to use on farm. I might just quickly take this opportunity to remind you about a couple of things. One, the handouts are there to download, and we'll have some others with those webinars around resistance management, so feel free to download those. This webinar has been recorded as well, so we will pop that up on the web page. We've also got, in addition to those resources off to the side, some USBs and other resources that can be mailed out if you are dealing with growers day to day. This has got all the fact sheets and videos and bits and pieces on it from the last couple of years of this extension project. But look, we've really stuck to time. I'd like to get everyone out of here on the hour and to continue on with the Friday afternoon. So thanks very much for attending, and we look forward to seeing you next time. Great, and thanks to Paul and Jessie.
Thank you.
Thanks everyone. Bye.